Testing. Okay, good afternoon, everybody, and thank you for coming to this week's departmental seminar. I've got a couple of announcements to make, and then I have the pleasure of introducing Todd to us today. So first announcement is that Larry Grabo had a heart attack 2 weeks ago, but he's good. So I didn't really want to send that out via email to everybody, but he had surgery. So he had a completely blocked lateral descending artery, and his body had developed some sort of bypass venation, which is why he didn't know he had any kind of heart problem. He had an event at work in Belgium, and fortunately his coworkers identified what was happening and were able to call immediately for the ambulance to come and pick him up. And I gather he's at one of the best hospitals in Western Europe. And so he's been in the hospital for the past 2 weeks. They identified the blockage, and he had minimally invasive robotic surgery on Wednesday. And I heard from his son that went really well, and the surgeon was very pleased with what he was able to do there. And so Larry will be in the hospital in Belgium for about another week, and then about a 5-week recovery. So we're hoping to be able to extend his sabbatical, since no one goes on sabbatical to have major medical issues. So hopefully he'll be able to kind of recoup that time on the backside. So bad news, but also good news. I think he's doing very well. So I'll let you guys know if there's any changes there. The second piece of information is that next week, instead of our usual Friday seminar, we're having the 50th anniversary of the long-term no-till plots. The vast majority of you should have seen that announcement. This is a different kind of event for us, and we've hijacked the Shirley Phillips Award in the process. This is not going to be a yearly thing, but because it's the 50th year celebration of these plots, we decided it made sense to kind of bring all that together. Most of you should have seen this and know what's coming. We've got 2 outside speakers, Matt Liebman from Iowa State and Dave Montgomery from the University of Washington are going to be speaking. And then John Grove is receiving this year's Shirley Phillips Award. And so he will be giving the presentation around noon or 11 o'clock, I believe, on Wednesday. Bob Blevins and his wife are making the trip to be here. So they have been living in Arizona for a very long time. Bob is in advanced Parkinson's, but their son is coming with them, and I think they're very, very excited to be coming and going through the celebration. In the process of planning this event, we're in the main room in the Good Barn, and we can't have it be everything. There's not enough room in there for us to have all of Plant and Soil Sciences, 150 people show up and still be able to feed everyone and do the seminars and do everything. So we haven't put a departmental-wide invite out. If you feel desperately that you need to be included in this and have not received the invitation, let me know and we'll see what we can do. Right now we've got about 60 to 65 people registered for the event. So I hope you can be there and make it. I know many of us will be coming and going as our schedules allowed, which is fine. At least 11 or 15 of our emeritus professors are planning to be there, and it's a pretty good sized deal to have Bob be able to come. I think Bob's in his mid-80s, possibly upper 80s. So it's a big deal for those guys to be there. So please try to join us for that. Any questions on what's planned for that day? Okay. And then the next seminar, so that's next week, will be on Wednesday. And then the week after that, if you can believe it or not, is Hannah's 2-year review. Time passes fast, doesn't it? So I will be circulating her 2-year CV the Monday after Thanksgiving. So you guys will see it and get it for about 5 days. And then she'll give her seminar. And so I especially request that all faculty be here and try to make it for her seminar and the discussion that'll happen then. And with that, do we have any other announcements of any kind that I'm missing? Okay. With that, it is my great pleasure to introduce Dr. Todd Pfeiffer to you. We had a cancellation in this program, and one of the negatives about participating in the 3MT when I'm sitting there is that I get to see all the interesting things you're doing. So you might remember Todd gave a 3MT about some new analyses he's doing on his sorghum data collected over his career. And so I thought it would be interesting if he would be willing to come and share some of that with us today. So we're thankful that he agreed to do that and is filling in at the last minute here. Many of you, who knows where Todd got his bachelor's degree, his BS degree? That's right. Very good. So he got his bachelor's degree with us. He's a graduate of the agronomy program back in 1977. He then ended up getting his master's and his PhD at the University of Wisconsin, and then he came straight back to us. So six years away, and then back to us. He joined us as an assistant professor in 1982, and he's been with us ever since. And most importantly, served as our chair for 2009 until 2017. His research program over the years has varied. He started out with us as a soybean breeder, did that from 1982 to 2009, when GMOs sort of totally changed the landscape of soybean genetics and ownership and how things were happening there. He made the switch at that point in time into sweet sorghum breeding, and he's been doing that pretty much ever since. He's been very active in our undergraduate teaching. As you all know, teaching is an important thing to Todd, and he spends a lot of time and effort on it. He taught genetics for a lot of years, PLS 104 and then 103 when it came in, and he did crop production in 1984 and 2009, or 2019 now. He did it for years in 1984, now he's back to it in 2019. I'll also have to say that I've heard Dennis Egley tell me that Todd was the best student he ever had in 502, isn't that what it was? Isn't that what you told me, Dennis? Todd also did an undergraduate research project with Dennis Egley on C14 and soybeans and trying to understand when soybeans basically stop developing anymore. So Todd has the status of an eminent undergraduate from our degree program here, and it's my pleasure to welcome him and have him show us about his sweet sorghum research.
Well, thanks, Rebecca, maybe. Yeah, so Wisconsin. Well, last week I was in Wisconsin visiting the grandkids, and when you know it, the two old people, Janet and I, both got whatever grandkids carry. And so if I start coughing, I'll just take a break. So some sweet sorghum stories. I was hoping Rebecca would introduce the title sort of like the courtesy car ad, some sweet sorghum stories. But these are some sweet sorghum stories. Okay, sorghum. Lots of different sorghums, all sorghum bicolor, but lots of different types, mainly based on height. So we have short grain sorghums and forage sorghums and sweet sorghums and very tall bioenergy sorghums, which are just sweet sorghums that are photoperiod insensitive, so they don't flower and they keep growing really tall. Lots of biomass. So lots of sorghums. I'm going to talk to you about the sweet sorghum crop. What I've been working on mainly is sweet sorghum for syrup, but some sweet sorghum for bioenergy. Sweet sorghum for syrup. It's a different crop. The crop's value is in the sugary juice in the stem. So we're looking for biomass and sugar. Process it through a mill to squeeze it. It's like a sugarcane mill, only lot smaller. But we collect the juice, take the impurities out, and settling in preheating tanks. You wouldn't want to put that on your pancakes, I don't think. But then it's cooked into a beautiful syrup. And sorghum syrup, mainly Appalachia, a heritage food across the South. It has its own flavor, just like maple syrup has its own flavor. Sorghum syrup has its own flavor. It's robust. This says I'll put it on a biscuit. You can put it on cornbread. I like it on sausage if you cook. Good in barbecue sauces. If it calls for brown sugar, just put in the same amount of sorghum syrup, and it tastes great. And so now we're all hungry and ready to leave. Yeah, well, I really like it. Old friend Morris Bitzer, he always had, every night he had sorghum syrup on ice cream, and it's good that way too. Kentucky, Tennessee, and North Carolina produce the most sorghum syrup, so that's why I got into it. I got into it with our old friend Morris Bitzer back in the early 2000s with the tobacco buyout program. University of Kentucky had a new crop opportunities grant program to find substitute enterprises for small tobacco farmers. And because we were active in sweet sorghum production, Morris and I went to work on that. Unfortunately, Morris died too soon, and he's not working on it. And I'm the last person still working on it. The goal, improve sweet sorghum varieties for use in syrup production. And I'm going to talk about these specific objectives and how we found the varieties to meet those specific objectives. A little aside on this picture. I don't remember this picture. I don't know when it was taken or where I was, but I was scouring the World Wide Web looking for pictures to steal, to appropriate borrow for this talk. And I got to steal myself. Because I found it on some random sweet sorghum website. I don't know why, but I'll be on the World Wide Web for a long time now as a sweet sorghum person. So beware where your pictures might go. You're not going to know. Okay, high yield, biomass. You can see those pictures. Sweet sorghum can get 12, 15 feet tall, one of the highest biomass producers around. Grain sorghum is better known, more widely grown sorghum. All grain sorghums are hybrids. Heterosis in sorghum is about 20%, so they make a perfect hybrid. When we started, sweet sorghum hybrids didn't exist. So if we were going to look for higher biomass, higher yields, we looked at the hybrids. And sweet, it worked out. Sweet sorghum produced the same hybrid vigor as grain sorghum. We made 12 hybrids and compared them to their pure line parents, and hybrids were 10% taller, fatter stalks, greater weight, and 22% more juice. And you saw juice is what we're really after. So great for hybrid vigor. So we tried testing a bunch of hybrids and producing a hybrid. We had this sorghum hybrid for use in syrup production in Appalachia, and it worked. So that hybrid was K.N. Morris. K for Kentucky, N for Nebraska, and Morris in honor of Morris Bitzer. So hybrids in sweet sorghum are use cytoplasmic male sterility still. Hybrids in corn a long time ago used cytoplasmic male sterility, but with the Texas cytoplasm and southern corn leaf bite, that crapped out. But still working in the in grain sorghum, and they still make cytoplasmic male sterility hybrids. So you need a male sterile line, sterile cytoplasm, and that same genotype and a fertile cytoplasm, so you can get pollen to keep recreating the sterile line. And the line we used as a female was N100, and that's where Nebraska came from, because that was a Nebraska line. And then the R line is a restorer line, because it has genes in the nucleus, which will overcome the sterility from the cytoplasm and give you a fertile hybrid. And so Dale was our male parent, the most popular sweet sorghum variety at the time in Kentucky, and we produced the K.N. Morris. But it's a little different because our R line Dale wasn't really a restore line, so it gave us some differences. There's lots of different male sterile cytoplasms in sorghum. A1, A2, A3, A4. You can see no yellow pollen from these sterile heads compared to from a fertile head. Our line from Nebraska, A3N100, used the A3 cytoplasm, and Dale doesn't restore fertility. Actually, almost very few sorghum genotypes restore fertility to the A3 cytoplasm, less than 1%. Grain sorghums almost all use the A1 cytoplasm, and about a third of sorghum genotypes will restore fertility to the A1 cytoplasm. That's why it's widely used in grain sorghum production. So we had a sorghum hybrid that, if it didn't get pollen from anywhere, was also sterile and didn't produce seeds. Okay, well, it produced 20% more sugar per hectare in our tests, and it didn't produce seeds when grown in isolations. We thought that was going to be good for lots of reasons. You didn't have this great big head on top of the sorghum weighing it down to pull it over. The syrup producers weren't going to have to come in and de-head the sorghum because they didn't have seeds, so they could still run the whole plant through the mill. And we thought we were not going to have competing sinks. No reason to send photosynthate to the seeds if the seeds weren't there. So that probably gave us a little more sugar in the stock. So we thought that sterile hybrid was going to be good for sugar production, syrup production. But at the same time, there was a biofuel craze going on. I stole this from Tom Keene. This is his intro to his hemp talks now. Hemp is happening and hemp is hot. Well, back in 2008, if I had known it then, I could have said, biofuels are happening and biofuels are hot. 2009, Kentucky had an energy plan that we were going to derive 12% of our motor fuels and electricity from biofuels, requiring 26 million tons of biomass a year. That's a lot of biomass. Southeast Biofuels promotes sweet sorghum in Kentucky. Their plant in Hopkinsville in 2012 was processing sweet sorghum into ethanol. Lots of farmers thought they were going to have energy crops in their rotations. There were companies, I won't pick on Ceres, but it makes a good story. They were energy crop companies. They were just doing a biomass breeding. So during that time, we were getting calls from people saying, well, we want to grow 1,000 acres of a hybrid sweet sorghum, and you've got one. Mostly this is in Florida or Louisiana, and they wanted to space it in between sugar cane because it could fill the gaps in with sugar cane. And they'd say, do you have enough seed for to plant 1,000 acres of K.N. Morris? And I say, heck no, I don't have 1,000 acres worth of seed just sitting around. But how would we produce seed if people wanted it? So this story is more about the seed production than it is about K.N. Morris. So both parents of K.N. Morris are sweet sorghum, so they're both tall. One in the front is N100, the one in the back is Dale. It's quite a bit different from hybrid grain sorghum, where the females and the males are both short. So the question is, how are you going to get that seed? Well, on the left, that's our first K.N. Morris production. You're not going to produce enough seed for 1,000 acres that way. So fulfilling the dream of using sweet sorghum for biofuel production, we had to go with somehow get to producing combine harvestable seed. Well, that meant our female line had to be short. Well, all sweet sorghums are tall. That's why sweet sorghums are useful, because they're tall. So we had to be able to create a short sterile line. There are 4 dwarfing genes, at least in sorghum, that makes it short. So our line is tall. We would look out here, and that cross a tall onto a short. All the tall alleles are dominant, and so your hybrid's going to be tall. So the key is, can we produce a short, sterile female for this hybrid? Well, those of us of a certain age remember my mama used to tell me, Rosanna, Dana, it's always something. Well, it's always something. When you have sweet sorghum heads that don't produce seeds, they can become infected with sorghum ergot. And that produces this sticky substance, real sticky mess. Well, we don't have sorghum ergot in Kentucky, so it wasn't a problem on K.N. Morris, but across the southeast and in the Gulf Coast, they have sorghum ergot. So you're producing thousands of acres of K.N. Morris in the southeast. This is what you really call gumming up the works. So we had to get away from the A3 cytoplasm. At the same time, we were creating a short female. So the goal was to create a short, sterile female with a fertility restoring male line. This isn't a unique idea of mine. Everybody who is crunching on this was going to do it the same way. But most people were going to look for grain sorghums, which were short, and use the same male sterile female grain sorghum parents to cross with the sweet sorghum. But I'm thinking, wow, we already got a proven sweet sorghum hybrid. Let's try to make N100 sterile. But would it work? Again, sometimes things in life just work out sweet. So tested N100 with the A1 cytoplasm sterile, and it was a maintainer. It could be the female. And tested Dale with the A1 cytoplasm sterile, and it was a restorer. It could be the male. I mean, it couldn't work out any better, couldn't work out any better. So 2010, started working on this, create a short N100 genotype in the A1 cytoplasm. So I had a short grain sorghum, A1 cytoplasm, crossed N100 onto that. And what are the progeny going to be? Well, they're going to be tall. Well, I don't want them tall, I want them short. They're going to be tall. But also, N100 is a maintainer, so they're sterile. Now what do? They're not going to produce seed. I can't self-pollinate them to make them short. So I got this tall sterile. So the other thing was to cross N100 onto a fertile grain sorghum. And that's also going to be tall, but it's going to be fertile, so I could self-pollinate it and find those segregating short sweet sorghums. And then I can use those to cross back onto the tall sterile F1 and try to slowly get that shorter in a sterile background. Well, it takes a while. Standard back crossing, goes 50, 75, by half each time you back cross, you get back to the recurrent parent. And so producing the maintainer line N100 to make it short. I could cross and self, cross and self, cross and self. I selfed in the greenhouse so I could get 2 generations in a year. And so it only take about 6 years. But then I always had to cross my sterile line back to this maintainer line. So instead of going 50, 75, 87 and a half, cross 50 by 50, and it's still 50, 50 by 75, only get to 62 and a half. So just doing them in parallel, a sterile one, a sterile A line, and a maintainer line, trying to get shorter and sterile and shorter and sterile as we go along. So 8 years from 2010, that's about now. So am I getting close? Yeah, I think I'm getting close. They're still a little taller than I like. They're about 1.6, 1.5 meters. I'd like them to be about 1.2 meters. But this year I made some crosses of Dale onto my A1 N100, 1.6 meter short female. And I'll test the hybrid next year to see if the new hybrid is going to work as well as K.N. Morris. So 2019, biofuel is happening and biofuel is hot. Well, not. Haven't heard much about biofuel. Well, freaking fracking changed our energy picture. All these, these are vertical well productions of oil and gas. I mean, horizontal well production. Horizontal wells and oil production now outnumber vertical wells. Horizontal wells are fracking. 2008, that energy proposal was both energy independence for the United States and Kentucky, as well as managing climate change, CO2. But the United States wasn't producing near enough oil. Now with fracking, the United States is the world's top crude oil producer. And so for energy independence, we don't hear anything about biofuels anymore. And political climate change. In 2009, we were going to pass cap and trade. The House of Representatives passed the Waxman-Markey CO2 cap and trade bill. Never passed the Senate. But at the same time, there were carbon credits being bought and sold on the Chicago Stock Exchange through Chicago Climate Exchange. Doesn't exist anymore. So as a result, the crop world also changed. 2007, Ceres raises $75 million to develop dedicated energy crops. Once again, I'm not picking on Ceres. It's just they were doing it in sweet sorghum, so I followed their story because they were a competitor. And then they had an initial price offering, and you can read the initial public offering of their stock, what they said. I like the in English. If biofuels and biopower do not scale globally, we are toast. Well, biofuels, they were toast. In 2016, Ceres completes shift away from bioenergy into forage and feed crops. So less than a decade, they went from this is the hottest thing to now they're back to breeding forage and grain sorghums for their business. But things could change if we adopt the Jay Inslee climate protection plan, or if President Warren bans fracking. We will have enough hybrid sorghum seed to corner the market again for the University of Kentucky, and lots of royalties for the department, right, Rebecca? Sounds great. Okay. But so much for dreaming. Back to sweet sorghum, another story, back to sweet sorghum for syrup. Objective was early maturity and high Brix. So syrup production is really just sugar in the syrup, boil away the water, and leave the syrup. So its value is sugar per hectare. Vegetative biomass times juice percentage times the Brix of the juice. That's what we're looking for. So we talked about the vegetative biomass with the hybrids. Juice. Juice in the stems is just one gene. Dominant gene is pithy or dry. You could heat it and it doesn't shrink any. The recessive allele is juicy, so when you heat it up, you boil up, get rid of all that juice and the stem collapses. I think Sharon sent me an article on this and said, this gene is what? One of Sharon's favorite transcription factors. I can't believe how many genes are transcription factors. That's just an aside. It still blows me away that they're not all structural genes, they're transcription factors, but single genes. So juice percentage varies not by much. It could be somewhat a function of our mill, because the most we can get out of that little mill I was showing you is about 50% juice. But we don't see much different in juice. So what about Brix? Brix is a way of measuring really soluble solids. But in sweet sorghum, most of those soluble solids in the juice is sugar. So when I say Brix, I mean sugar. I really measure Brix, but I convince myself that I'm measuring sugar. So Brix, it's polygenic, so we can breed for higher Brix. You read it in the hydrometer, and you can see what the Brix level is. Once again, when we started, most early maturing varieties are heirloom varieties from the 1800s. So sweet sorghum was a big sugar crop in the US in the 1800s. It's going down, down, down as we started importing sugar. And the most improved varieties were late varieties. Why was that? Well, the only sweet sorghum breeding station was the USDA Sugar Crops Research Station in Meridian, Mississippi. And so those varieties aren't late for them, but they're late for us. Sweet sorghum is grown from Wisconsin to the Gulf of Mexico. Well, they can't grow these varieties in Wisconsin. So started working with the National Sweet Sorghum Producers and Processors Association. At the time, Dr. Bitzer was the executive secretary of that organization, so we were working closely with them and trying to get earlier maturing lines for Wisconsin, Pennsylvania, as well as to smooth out the production stream in Kentucky. But these were all the heirloom varieties, and they had the lower Brix. So that's the question: is late maturity required for high Brix? Could we have early lines which were high Brix? So did this experiment. We made 6 populations, early by late populations. Out of those, collected the five early lines, 5 mid lines, and 5 late lines. That was 90 entries, and tested them at Quicksand in Lexington for 2 years. And sweet, high Brix doesn't depend on the late maturity. When they're in the same background, it didn't matter. But then we went looking for a variety which was early maturing and high Brix. And a couple of years ago, we released one, Kentucky 238. Parents were Simon and Keller. Doesn't mean anything to anybody in this room except me, so I don't know why I put it on the slide. But Simon's very early and Keller's very late. 238 is Simon maturity. It's very early. Simon and 238. 238 has the highest Brix, any test I do, anytime, it has the highest Brix in the very early background. And you can see that here. A couple of weeks earlier than our good late lines. Okay yield, but the highest Brix, 2 to 3 percentage points higher, and adequate sugar yield. So hoping that northern sweet sorghum syrup producers can use this line in their production. So back to Rosanna. If it's not one thing, it's another. Tim can tell you in plant breeding, if it's not one thing, it's another. If it's not one thing, it's another. The bane of sweet sorghum producers is lodging. You get that 15-foot tall plant, you get that big seed head on top. Half the time you get a storm coming through after it's headed and set seed, and you get lodging. You might be able to see these pink ribbons in here. Those are ones earlier in the summer I had selected because they looked really good to me. Well, mistake. So I didn't harvest them, but the next objective is to reduce lodging. So I made a bunch of crosses with this breeding line 54. It's a tall sorghum. It's really attractive. It has thick stalks, but it has dry stems. Cross it with a bunch of standard sweet sorghums. One of them was Theus. And so now I have this line KY13644-2. It has thick stems, and Seth Bolt's group with Norbert, they've looked at it a bunch and says it's really high in lignin. That's the exact opposite you would want if you're Chris or Ray looking at forage sorghums where you want really low lignin. But here, high lignin is good, and it's lodging resistant. So another one of those maybe making lemonade out of lemons. I don't know. So in 2013. Early in September, we had this big storm, and I had all those plants tagged, and a big storm blew 75% of them down. But there was this one row in the field, nothing blew down. That happened to be row 644. That's where I cleverly named this, you know. And so I hadn't tagged any of those plants because they were too short. But they didn't fall over. They couldn't be blown over. So I harvested a bunch of them, tested them the next year, and the best one out of that was plant number 2. So that's where KY 2013, row 644, plant number 2. That's what we're looking at here. But it's late maturity, it's short, and it's lower Brix. Never would have chosen it except for its lodging. Show these data to the sweet sorghum producers and processors, and a bunch of the growers are interested because if you get lodging like in those pictures, they can't harvest their crop, and they'll do anything not to have lodging. So it's not really that great. It's late, it's short, slower yielding, doesn't have great Brix, but the producers are interested in it. So we tried a little participatory plant breeding. I sent seeds to as many of the producers as wanted to try it out, and I think it was 12 of them. They were growing it this year to see if it lodged in their environment, and more importantly, to see if it made good syrup, because I don't have the ability to make good syrup. And hear back from them whether they like it or not enough that the good lodging resistance supersedes the other lower quality traits, but we'll see. Okay, those are some of my sweet sorghum stories. So if you want to know anything else about sweet sorghum, I'll try to answer it for you.
Any questions from the crowd?
So, Todd, how you feeling?
I didn't break out coughing, so I'm feeling pretty good.
Do you know anything about the heritability of the low oxygen trait?
No, I don't. I don't know how many genes there are because I had crossed, breeding line 54 with a bunch of other sweet sorghums, not just this. And it was only one row that looked really great. And there were a bunch of other rows in the nursery that had a background with breeding line 54. So I would guess it's pretty low, but I haven't done any genetic experiments to tell you.
Are there other breeders of sweet sorghum in the U.S. or
Other countries, yeah, India, Africa, they're used a lot. So there's at the international stations, there are sweet sorghum breeders. I don't know of any others in the U.S. since the collapse of the biofuels market.
So you mentioned that lots of this line
I'm waiting to hear back. One person whose granddaughter is in the class that Tim and I are teaching. He made some and brought it to me, and it tasted good to me. He thought it was a little bit. He thought it was a little bitter, but I couldn't tell. It was a.
What kinds of quality do they measure quality in sweet sorghum syrup?
Do they measure quality? It's all personal preference. But every year at the
Professional tasters?
they have all the producers are professional tasters. No, really. Every year at the National Sweet Sorghum Producers and Processors meeting, all the producers can bring in a sample of their syrup for that year. And they have some qualities it has to have. It has to be 78 Brix or higher, and some other things. But then they recruit from the audience 6 people to be tasters. And so they taste 50, 60 sweet sorghums and rate them. And as long as your high blood sugar doesn't go up, you get to taste all 50 of them. I've done it before now as a non-professional taster, and you can taste differences, but it's personal preference. For the mass market, you'd like it really light, like the syrup I showed you. For the heritage market, people who've been eating it families have been eating it for decades. They like it a little darker and a little bit more bitter, with a bite to it. So it depends on who the judges are. But K.N. Morris won one time, and KY 1810 won one time. So our varieties are good enough for syrup production. Some sweet stuff, yeah.
Has anybody thought they can list yet?
I would bet they have, but I haven't had the pleasure of trying it. So.
Okay, and you may have said this, but I'm making, what is molasses?
What is molasses? Well, molasses is a byproduct from sugarcane processing. So after they processed it, and I don't know exactly where in the line, but molasses is even darker and more bitter than sweet sorghum syrup. So there's a big debate with the producers and processors. So there's a big push. You can certify your sweet sorghum syrup, because the other problem is people would cut it with corn syrup to make it lighter. And so you have to be 100% sweet sorghum syrup. And you sell it as sweet sorghum syrup. And you don't call it molasses. But people, once again, in these Appalachian, southeastern communities, everybody calls it molasses. And so now when they're selling it to their friends and audiences, they call it sorghum molasses to differentiate it from sugarcane molasses. Yeah. Onto. Why? I don't know. And it correlated. Why do they have more sugar? I mean, there's a lot of differences in sorghum. I didn't want to use two. I was going to show this slide, but I didn't because I want to have enough time. But grain sorghum somehow, some reason, accumulates its energy in the seeds, where sweet sorghum B accumulates its carbon in the stalk. We don't know what genetic change is different there to change the partitioning. Sweet sorghum is different than sugar cane and how it loads the sugar into the stem and how it processes the sugar. But Monsieur, I can't tell you any physiological reasons behind that. Wheat.
Speaking of opportunity farmers, any progress in the study of variety of drought resistance for this
drought resistance. Well, we haven't looked at drought resistance in sweet sorghum. Mostly that's grown in the southeast. Grain sorghum itself is a more drought resistant crop. It's primarily, the U.S. is the largest producer, but that's all Kansas, Oklahoma, and Texas. So where you can't grow corn, they grow grain sorghum. And so it's naturally drought resistant at some point. I haven't read enough on grain sorghum to say what's the range in drought resistance, but I know people work on that a lot. So I would expect there's a range in differences.
Do they do anything with the stalks, with the leftover, once they get the juice out? What happens with the rest of the material?
Mostly it goes to animal feed because the sorghum producers aren't really big. About the biggest I know is like 65 acres, which is big, but not thousands of acres. But for bioenergy, when they're going to use it for bioenergy, it's really a three-phase process. They save the seeds, and they make ethanol just like they do with corn. They take the juice out of the stalks and they make ethanol, and then they would burn the stalks for coal firing for electric production. That's how they do. The Brazilians could tell you the story better. That's how they do sugarcane. A lot of sugarcane processing plants are totally energy independent because after they process out the sugar, they burn the cellulose and make their own electricity. Who's going to continue this work? First person to talk is a volunteer, right? You can do it. Yeah, you can join me next summer, and then it's all yours.
Along those lines, we got a form from HR that said we're retiring next month. I don't know if you told you that. You had to send it back. I only had the wrong year.
Oh, well, thank you. At least you didn't turn in the form telling me I was retiring next month. Wow. Okay.
Yeah, some things are better not. Yeah, David.
So the very dark color is probably all mostly that could be useful and helpful, right?
Yeah. So people who want to promote selling sorghum syrup, yes, they say it's a good health food. But you have to eat a lot of sugar. I don't say you can get all these antioxidants if you eat a whole lot of sugar. But yeah, is it phenolics? Well, it's different things. It could be phenolics. It could be they heat it and burn it. Some of it, they say that sorghum accumulates nitrate in the stalks, and if you get too high nitrate, that gives dark syrup. So they don't like we don't use much nitrogen on sweet sorghum production because they don't want nitrates in the stalk because they say it gives them dark syrups. But a lot of it is phenolics, tannins, things like that.
When they're producing syrup, if you pretty strongly that the lime makes a big difference in terms of flavor. Does the cooking process make a difference? Are there different cooking methods that folks use when they're making the syrup?
Yeah, I mean, different ways of cooking. So this is a big cook, and so they have steam tables and it's continuous flow. But really, most of them are the same. You're just boiling off the water, skimming off any of that little green chlorophyll and other things that didn't settle out in the chilling and preheating chambers. But the people who do this, they say it's all in the timing. You got to pull it off at the right Brix before it gets dark, and so it's all in the timing. But basically, all the processes are boil off the water. So when I try doing it on a small scale, we do it in a microwave. A small amount you put in the microwave, heat it for however long until you get to the right stage, and then you take it out. So cooking it's about the same.
Do you have a favorite brand? Can you put in a plug for your favorite? What producer in Kentucky? What's your favorite? You told me, I think you told me.
Well, it's more where can you find it? So I get the Randall Rock sorghum from Windy Corner, or I get Danny Townsend sorghum for Reed's Valley Orchard. And unfortunately, you can't find it at Kroger's. I go to the sweet sorghum meeting and I get a little graft there, so I bring a couple jars home from. And but those are two. One's in Montgomery County, one's in the Woodford County. Those two sell around here. I like them.
What variety are they growing, do you know?
Well, they grow a range because they want to harvest from very early to very late so that they can process over about a 1 month period. If you only had one variety, it's all mature at the same time, you can't process it fast enough. So they would grow all the way from early, like 238, to really late, like M81E, which once again doesn't mean anything to anybody in here. But over a month difference in maturity, so they can make sorghum over a month.
You might ask Princeton if they have any questions.
Princeton, any questions?
No questions here.
Okay.
Have we got one more for Todd, or are you ready to say thank you very much? Like
well, thank you very much. Thanks.
Yeah. I just submit. I received